Dear delegates, we will be starting the scientific sessions exactly at 11:30 a.m. The first will be a keynote address by Professor Ralph, followed by invited talk by Professor R. Umashankar, and we part for a lunch at 1 o'clock. 1 to 2 is lunch will be served in the Botanical Garden of US Bangalore, and exactly 2 p.m. we will be starting the technical sessions, and there will be two lead talks in the technical session 1. First one by Professor T.S. Suryanarayanan and second one by Dr. A.K. Saxena, which will be followed by the session talks by Batina Purnesha, K.P. Girivasan, Lakshmiti Kumar and Maria Johnsy. And at 3:30 we are planning to have the tea break, 3:30 to 4 p.m. And technical session 2 is proposed to be at 4 o'clock. We'll be starting by 4 o'clock, 4 to 5:30 p.m. And there will be one lead talk by Dr. Pius Thomas and followed by session talks by Meenu Thambi, Elizabeth Jose, R.R. Vangude and Sanjay Kumar. And 5:30 to 6:30 is the time for poster presentations, and 6:30 to 7:30 p.m. is the panel discussions. And all those who are presenting their posters can hand over their e-posters to Miss Pratibha next to the registration counter. And all those who are presenting the oral presentations can hand over the soft copy of the presentation to Mr. Tinu Thomas here. So once again I request all the poster presenters to hand over your e-posters to Miss Pratibha near to the registration counter.
Yes.
Just
okay, great.
wait. 11:30.
Okay. My first slide is this one. Yes, makes sense to me. I know this slide. So I move that forward here?
Yeah, you can do that, or you can use this.
And pointer is here.
That's pointer.
Okay. Too much technique is too much for an old man.
I request all the participants to be seated. In few minutes we will be starting the scientific sessions. So it's exactly 11:30. Dear delegates, welcome back. Now we are going to start the scientific sessions of the symposium, and I wish you to have a fruitful time spent ahead listening to science. So first we have the keynote address by Professor Ralf Ollmüller, Friedrich Schiller University, Institute of General Botany and Plant Physiology, Jena, Germany. I request Dr. Nataraja Karava, the symposium convener, to welcome Professor Ralf with a brief biodata.
Good morning. I am here again to welcome our collaborator friend and excellent researcher who is a Professor C4 Chair Plant Physiology at the Institute for General Botany and Plant Physiology, ENA, Friedrich Schiller University, Germany. I am very happy to announce that he is adjunct professor at US Bangalore. As I mentioned earlier, he was the Alexander von Humboldt Fellow at Washington University, Riken Fellow, Tokyo, Japan, and is the Vice Coordinator of International Max Planck Research School ENA and Group Leader in German priority programs on plant-microbe interaction, and also Group Leader in several German research priority programs. He was the Dean of the Biopharmaceutics faculty of Friedrich Schiller University, and he is the President of International Society of Endocytobiology. Again, he is re-elected this year as a president, and he was a president earlier, and Professor Ralf is the editor of Endocytobiosis and Cell Research. I am very happy to welcome Professor Ralf. Professor Ralf obtained his PhD in botany under the guidance of well-known Professor Hans Mohr, and his main research interests are plant signaling, plant endophyte interaction, and has published many research papers in high-impact journals, to name a few: Plant Physiology, Plant Cell, and many more. Currently, he is using Arabidopsis system to understand the communication between plant and endophytes. I am very pleased to welcome Professor Ralf for this keynote address. Professor Ralf, please.
Okay, can you hear me? Yeah. Thank you very much for the nice introduction, for everything that you are doing for me here to make my stay beautiful. What I would like to do today is to give you a brief overview of what's going on in my lab, and I'm trying to address a few questions. Which might be helpful for bringing together basic science and agricultural application. My group is working on the interaction of endophytic fungi with model plants, which can be used for the analysis at the molecular level. And all the data that I'm presenting you today are mainly concentrated on an endophytic fungus called Pyrethromospora indica. It's from India, as you can really understand from the name, and was isolated by Professor Ajit Varma 25-30 years ago, and now it's spread all over the world and many people are working on it. I briefly give you some ideas about Mortirella hyalina. It's another endophytic fungus. Both of them are interacting with many different plant species, also crop plots. Many people are working with them, and what I'm trying to do is to understand what is the mechanism of the symbiotic interactions. So this is just an overview to give you a small introduction. Of course, our goal and our strategy is probably the same. We are looking for endophytes which promote growth, more crop yield, better resistance, and if you go through all the available databases or data banks, you find about around 10,000 fungi that can be used for colonizing plants that have either, most of them have beneficial effects. I only have chosen those ones which have beneficial effects. And what we really have to do is to understand what is the mechanism of how these organisms interact with roots and why do they promote plant performance. So our basic understanding is that it makes first sense to test many of these different fungi in ecological systems, agricultural systems, ecological systems, to test whether they are beneficial for the plants. And then when we have a few fungi that really show beneficial effects, we can go ahead in the lab mainly to understand the molecular basis for this process. And for this we can actually use model systems. I present you a few data on Arabidopsis, Chinese cabbage, don't talk about barley rice. I will take you through these different aspects. As an introduction, I would like to give you my view of how the interaction may occur. And I'm looking both at the fungal side as well as the plant side. So my belief is that fungi have different strategies. So they can release hormones, different types of infochemicals, soluble chemicals, volatiles, toxins, effector proteins, and they can all be very different depending on the fungus that you're looking at. And all these compounds somehow have to influence the metabolism, the signaling events in the plant. However, a plant has only a limited repertoire to respond to these signals. The response is mainly determined by developmental programs, which are determined by the genetic background of the plant. And in particular, since endophytes can interact with many, many different plant species, that is a typical feature of endophytes in contrast to mycorrhizal fungi. So there can be no specialization. So they're interacting with one plant species and with another plant species, and they have to use the genetic repertoire or the plant developmental programs which are available in these plants that they are interacting with. So that means they can only manipulate processes which are established in a plant. And I think I have a nice example to really demonstrate that. And this is shown here. Again, for the beneficial fungus Piriformospora indica, interacting with the model plant Arabidopsis thaliana that we are using often in the lab, as well as Chinese cabbage. And you can see here the control roots which are not colonized. And here you can see the roots of Arabidopsis and Chinese cabbage when they are colonized by the Piriformospora indica. You immediately see a big difference. Whereas in our Arabidopsis, you have a little growth promotion, lateral root development, root hair development. That is about 30% increase in fresh weight. In Chinese cabbage, you see a dramatic stimulation of root development. You get this bushy root hair phenotype. And this is caused by a component released by Piriformospora indica into the medium as well as into the host cells, and that's a precursor of auxin. So that's a precursor that's released, and then the plant takes this precursor and synthesizes auxin, and the available auxin then in Arabidopsis can only promote root development to about 30%. You can show that. You can directly apply auxin to the roots and you see the same outcome as compared to what you find when you use Piriformospora indica colonization. However, if you do the same experiment with Chinese cabbage, Chinese cabbage is much more sensitive to auxin. And if you apply auxin to the roots, you have the same phenotype as with Piriformospora indica, which tells you the response of the plant is determined by its developmental program. Chinese cabbage has been bred in the last 180 years, and the whole goal of the breeding process is to produce more biomass, make bigger plants, and so on. This pasture has never been on Arabidopsis. So this plant is extremely sensitive to all signals that promote growth, development, biomass production, and so on. And it therefore responds very sensitive to the auxin signal from Piriformospora indica, whereas Arabidopsis does not have this history and therefore is not showing such a strong growth-promoting effect. Okay, what I would like to do today is just touch 5 topics, all related or all directly interfering with the interaction of endophytes with roots, different model systems. And let's get started with, I think, one of the most important questions. How do the two organisms recognize each other? What is the requirement for the interaction? And I think one common feature is that the fungi, when they approach the roots, they release some chemicals even before there's a physical contact. And this chemical induces calcium elevation in the cytoplasm of the root cell. I think this appears to be a very common feature. I don't have found an exception yet. But what is very interesting is that this calcium elevation is mediated by quite different chemical mediators or chemical compounds from the fungus. I tell you a story today about Piriformospora indica, which is using cellotriose in nanomolar concentration to trigger calcium elevation, and what we can learn from it. And a completely different story that I'm not talking about today is Mortirella hyalina, who's using this organism is using a different chemical to induce also calcium elevation, and that is genetically, both in the plant as well as in the fungus, completely different. So, calcium elevation, you can see here. Why is it so interesting? Because it's very rapid. This is a minute scale. So when you apply extract from the fungus at time point 0, the maximum calcium elevation occurs after 90 seconds. So if you want to find out whether a microorganism is interacting with the roots or with the leaves maybe also of a plant, a very fast and easy readout is to check whether there's calcium elevation. Yeah, here I show you the two fungi that we are working with, Piriformospora indica, Mortirella hyalina. The pattern looks very similar. After 90 seconds, you have maximum level of cytoplasmic calcium in the host cell, and then the level declines again. If you take a control fungus like yeast, which is not properly interacting with roots, you don't find this calcium elevation. So this is a nice readout, a nice marker to really show that an interaction is going on. There are many questions related to this calcium elevation. One is, and I think that's the most important question, is this really important? Is this just calcium elevation, or is this calcium elevation really necessary for establishing the symbiotic interaction? And I hope I can convince you in the next few minutes that without this calcium elevation, you don't have an interaction, no proper interaction. So let's talk a little bit about a little bit complicated story. I hope I can bring that over to you easily with the next few slides about Piriformospora indica, the chemical compound that is released by Piriformospora indica to induce this calcium elevation. So, this you have seen already. We have this calcium elevation after 90 seconds. If we use an extract, you can use different types of extract from the fungus. You can either grind it or enrich use an enriched fraction. And we use these fractions to purify the compound that induces calcium elevation. That sounds very easy, but from this part of the slide to this part of the slide is about 5 years' work of 2 PhD students, because the compound is present in nanomolar concentrations. You need a huge of material to really purify it. So we purified more and more fractions and always tested calcium elevation, and then ultimately when we had one fraction that contained only one compound, we determined the chemical structure, and it turned out, this is elucidation of the structure by the classical techniques, it turned out that is cellotriose, a very simple compound, 3 glucose moiety, and the cellotriose, if you have a lot of money, you can buy it, it's expensive, and it tells you that like the exudate from the fungus, also the cellotriose that you can buy induces this calcium elevation. And what is very important for the next part of my talk is we identified an Arabidopsis mutant which does not show this calcium elevation. So we isolated a mutant from Arabidopsis which does not respond to cellulose and does not show this typical calcium elevation. We call this mutant SYCAM mutant, cytoplasmic calcium elevation mutant. I talk about this a few times in the next few minutes. So basically, no response to cellulose. So we characterized the cellulose a little bit more in detail. We found out, of course, it's dose dependent. That means the more cellulose, the higher is the calcium response. We checked whether there's some specificity for the triose form, which is the case. So if you use longer and shorter glucose forms, so bios or quattros or whatever, the response is less. 7 glucose moieties do not give any response anymore, and glucose alone also does not show calcium elevation. So there's quite a good specificity for the triose form, which shows that there must be a perception system which is somehow specialized in perceiving the triose form. And the triose form actually is doing something in the plant. And here comes also the mutant into play. Under certain conditions, it actually promotes growth. So this is cellotriose-treated Arabidopsis seedlings. These are cellotriose-treated Nicotiana seedlings. Compared to the control, there's some growth promotion, not always, but under certain conditions. More importantly for us is within half an hour, you have an induction of a number of different responsive genes, which are induced by cellotriose. That occurs in the wild type, but not in the mutant. And the response is very fast. After half an hour, you have a response, and then later on, the upregulation of the corresponding mRNAs is no longer visible. And we have a release or a generation of reactive oxygen species, not dramatic amounts of reactive oxygen species, but low amounts of reactive oxygen species which appear after 10 minutes after cellotriose application. So there are a few responses which we can measure, and the nice thing is we can use our mutant, which does not recognize cellotriose and does not induce calcium elevation, as a control. And this really tells you the calcium elevation is necessary for elucidating or for activating the downstream responses in the plant. What did I do? Okay. This slide only wants to remind you that the whole story is for Piriform spora indica. So we did the same with another fungus, Motirella hyalina. There's also a chemical compound, also present in very low concentration, also released by Motirella. It also induces calcium elevation, but it's not cellotriose, as a methylfuran. So it's a very different compound. It's genetically different in the fungus, and the response also in Arabidopsis is completely different genetically. Mutant, which do not respond to cellotriose, show no normal response to here, and vice versa. So that tells you there are many different infochemicals which are released by these microorganisms in order to stimulate calcium elevation in the roots. So how does the cellotriose affect plant performance? Is it really necessary, or is it important, or is it only an observation in the laboratory, or is it really relevant for the symbiotic interaction? So first of all, we figured out that is a response that is pretty much specific for the root. If you compare the calcium response in the roots with the response that you can see in the leaves, the leaf response is only 10-13% of the root response. So the specific root response to this cellotriose. And we showed that this is not only specific for Arabidopsis, but for many different other plant species as well. I told you that we have this SYCAM mutant, which does not show the calcium elevation, and we try to identify the gene that is mutated in this mutant. We use map-based cloning, Illumina sequencing, and whatever. Took also a lot of time to really pinpoint down the point mutation, and I don't want to go through all the procedure. And we found out that the mutated gene is a poly-A ribonuclease. That is, for you, first of all, not very spectacular, but it's interesting because there's only one gene in Arabidopsis and also many other plant species. So a poly-A ribonuclease recognizes messenger RNAs. They have a poly-A tail, and for some of these poly-A tails become degraded, shortened by this poly-A ribonuclease. Not all of them. Only about 1%, some people believe only 0.5% of all the mRNAs, the poly-A tails of the mRNAs become shortened by this poly-A ribonuclease, and then the mRNA will be degraded, and consequently products are missing, and one of these products must be responsible for the increase of the calcium elevation. We couldn't identify yet the target of this poly-A ribonuclease, but nevertheless we have a mutant in hand that we can use nicely as a control to show specificity of the calcium response. I come to that in a moment. This poly-A ribonuclease is really necessary for the calcium elevation. If we put the gene of the poly-A ribonuclease back into our mutant, Arabidopsis mutant, it responds to the fungus again, whereas the mutant does not, telling you that we really have a target gene that is absolutely necessary for establishing beneficial interaction between Piriformospora indica and Arabidopsis thaliana. Okay, what is the Celotrios doing? That is, of course, an interesting question. We did all the classical stuff, transcriptome, metabolome, and whatever analysis can be done. Basically, here's some sort of summary. It activates a small innate immune response, a little innate immune response, which is not comparable to what is happening with pathogenic microorganisms. It's maybe 10% of it. But this immune response is necessary for the symbiotic interaction. So we find genes which are upregulated, innate immune responsive genes. We find a little bit ROS production, cell wall thickening by callose deposition, and all of these genes and processes are different from what we know from pathogenic microorganisms, means chitin and so on. And all the genes and all the components which are required for the symbiotic interaction are not really involved in that. So what is actually happening is, that is shown here on this slide, one of the typical feature is that Celotrios controls colonization of the roots. And that is shown here. If we compare the mutant with the wild type is black and mutant is red. After 50 days of colonization, you find twice as many hyphae associated with the roots as compared to the wild type. So that means the plant cannot really control root colonization anymore when the Celotrios signaling does not occur. And that means it's very important to establish a certain balance between beneficial and non-beneficial traits in the symbiotic interaction. So there must be a certain degree of root colonization. If the roots are over-colonized, the benefits are somehow gone. So this balance must be established to really see benefits for the plant. And Celotrios is one of the players in this scenario. I don't want to go too much into detail because the time is already proceeding, but I would like to tell you what we can do now to understand how is Celotrios actually acting. So let's start with this cartoon that shows you what's happening if I give cellooligomers or Celotrios to a plant cell. So somehow there must be a receptor. Somehow there must be a calcium channel that is opened, because after 90 seconds we see elevated calcium elevation in the roots. Somehow there must be calcium-binding proteins which activate an NADH oxidase to produce reactive oxygen species. And somehow there must be signaling events which change gene expression. That is all that we have measured, of course. And this is all in a scenario, this is within 90 seconds, and this is briefly after 90 seconds. Gene expression changes occurred in 30 minutes. So if you compare all these events, then these are all phosphorylation events. Doesn't matter whether you look at mammalian systems, yeast, plants, all of these proteins which are marked here with a star, they are all either phosphorylated or dephosphorylated to be activated. And what we try to do is to understand which of these proteins become phosphorylated first after cellotriose application. So what we can actually do is we can look at phosphorylated proteins before the calcium peak occurs, and we can look at phosphorylated proteins after the calcium peak, and then compare that with the mutant which does not respond to calcium, does not show this calcium elevation, and can identify specific proteins which are very early phosphorylated, and those ones which are phosphorylated after we have a calcium elevation. And if you do this, then we can see there are two different pathways for a pathogenic stimulus as well as for cellotriose. Here, this is a defense process. So chitin is a component that is recognized by receptors, then it produces massive reactive oxygen species, ultimately activates MAP kinase signaling to defense gene activation. This all does not occur in the cellotriose signaling pathway. We have a receptor, we have then abscisic acid signaling, and we have then components which are activated in the primary metabolism, in particular in the sugar metabolism. And this tells you that we really have a stimulation of the primary metabolism along with the control of root colonization during these early processes of the recognition of the two symbionts. So I hope I could convince you with the first part of my talk, the other ones are much smaller, that the piriformospora indica releases a chemical compound that is present in nanomolar concentrations. We think it's present, we know that's present that's a cellotriose. You can barely find that in the soil. It's very difficult, also in the rhizosphere. It's very hard to detect. Actually, normally, even with our sensitive MS methods, it's too low to be detected. This is somehow specific for piriformospora indica because Mortellella uses a completely different system. And this cellotriose induces calcium elevation in the root cells, and this calcium elevation is necessary for establishing the symbiotic interaction. So far, this is what I wanted to tell you about recognition. I think calcium elevation is a very common feature of symbiotic interaction, and one can actually look at components which are involved in elevating the calcium level in the roots. Another aspect that I think is quite important, and these other stories are much shorter. So I think, we believe that resistant genes are targets of piriformospora indica, and for this we used an orchid, Oncidium, and this is a story that was initiated by Wei Ye, actually in China, and he came up with the following story, which I think is a very nice story. He's working with this orchid, Oncidium. It's very popular in China. I don't think it's very popular in Europe, but many people buy that in China, also in Taiwan, Japan, and so on. And it's propagated over many, many years, yes, and it has all the same genetic background, the propagation lines, and so everything there that you can sell has the same genetic background. And the problem with all this propagation is that this organism is very sensitive to Erwinia attack. Erwinia is a pathogenic bacteria that within a few days kills the entire orchid. It's a big problem in horticulture as well as in the propagation, as well as if you have this orchid at home. The other big advantage of the orchid is that the roots get very nicely colonized by Piriformis spora indica. And what Bayer did is he actually found out that colonization of the roots by Piriformis spora indica almost completely prevents the Erwinia infection process. So you show here's a control plant, yes, and when you use, so what he's doing actually, he is using Erwinia and putting the bacterium here on top of one of the leaves. And within a few days, actually 2-3 days, the bacterium and the information of the infection is going down to the root levels and then distributed into other parts of the plant, so that ultimately after 7 days the plant looks like this. And this process is almost entirely prevented when the roots are colonized by Piriformis spora indica. So the question is why? And he had a good idea that this could be, the reason for this could be that resistant genes are targets of Piriformis spora indica. And we had some preliminary data which supported this idea. What he actually did is he identified 99, that means almost all resistant genes in the orchid by bioinformatic tools, and the expression level of these resistant genes is controlled by microRNAs. So these are small RNA species which have a complementary sequence to the resistant genes. They both can come together because they have complementary strengths, and then the R gene messages the resistant gene transcripts are degraded, and there's no more resistant protein to be synthesized. So he did all the bioinformatic, and I probably will only show you the summary slide on that. What came out is the following story that is published. If you want to have details on the bioinformatic, you can go to that paper. What is happening is you have Erwinia infection here in the leaf. The information travels down and infects then the other leaves. If the roots are colonized by Piriformis spora indica, this process is blocked. Yeah? And the reason is the following. After Erwinia infection, the plant stimulates the expression of a number of resistant genes. Here are these resistant genes which are upregulated. And that's not very helpful for Erwinia. So what Erwinia is trying to do is to repress these resistant genes. And Erwinia is doing this by stimulating the expression of microRNAs. So these microRNAs recognize these resistant genes, form dimers, and then the mRNA is degraded and the resistant proteins can no longer be made. So that is the mechanism how Erwinia tries to propagate in this orchid. So if the roots are colonized by Piriformis spora indica, so signals from P. indica directly counteract this mechanism. So they strongly restrict accumulation of these microRNAs, and therefore allows accumulation of these R gene mRNAs. And this must be a very efficient mechanism, since the plants, as I've shown you at least 2 times, the orchid plants really stay green. It's more or less a yes or no answer in this system that he has established. This is interesting for several reasons. First of all, these are the R genes that he has identified to be really involved in establishing the resistance. But more importantly for me is, so there must be signals which are traveling from the root to the leaves, signals initiated by Piriformis spora indica to really affect activities, expression levels, metabolite profiles, or whatever, in the leaves. And that is really a task to understand how that can happen. So I hope I could convince you in the second part that there are resistant genes in orchids which try to prevent the spread of a pathogen, in this case a bacterium, And this mechanism is inhibited by Piriformospora indica by the regulation of microRNAs, which prevent the accumulation of the corresponding AS gene messages. So I think that's another mechanism that requires systemic information flow from the roots to the leaves to protect the leaves against pathogen infections. Now I will briefly touch a completely different story that is mainly done in the laboratory of Professor Kai-Wu Nye in collaboration with us. It started in Kai-Wu Nye's lab. And these are effector proteins which are present in the genome of fungi, in the genome of all fungi. And I'm talking only about Piriformospora indica. And the effector proteins appear in different fungi and appear to be very, very different. Nobody really understands whether there is some hierarchy, how they are related, and so on. I just tell you a story for Piriformospora indica, and I'm sure if even that is not understood, and I'm sure if you look at other endophytes, there will be completely different stories, completely different effector proteins that can all be used for different purposes. So the background of the whole story is Piriformospora indica is sequenced. Okay, so we have about 12,000 genes, and if you look at the genes which code for proteins, you have roughly 5,000 protein coding genes. That is comparable to other fungi, other root colonizing fungi. And out of these 5,000 proteins, about 400 contain sequences so that the protein is secreted in the apoplastic space out of the cell of the fungus. And from these secreted proteins, about 250 are small enough to have effector functions. So effector functions means these are small proteins which are secreted by the fungus. These small proteins go then to the root cell, will be taken up by the root cell, and then in the root cell can manipulate the metabolism somehow. But nobody has an idea what these proteins are doing. Actually, only one effector protein was investigated so far in Piriformospora indica. And he actually had a good idea. He said there are 250 effector proteins, potential effector proteins. Of course, we cannot work on all of them. The idea is which of the genes for these effector proteins are upregulated in the fungus when they are interacting with roots. I'll show you here a few examples. This guy, for instance. So the gene for this effector protein in the fungus is not expressed. But as soon as the fungus interacts with roots, you see a stimulation of the expression. Same here. No expression in the root, no expression in the fungus when the fungus grows alone. When the fungus interacts with the roots, you can clearly see that the mRNA goes up. Here are several controls and so on. So not all of the 250 effector proteins are probably involved in recognition or symbiotic interaction, but these are candidates which may probably play a role because they are only expressed when the fungus interacts with the roots. So he then did all necessary studies to really show what's happening with these effector proteins. We have chosen 39 effector proteins, overexpressed them, analyzed them. Here are a few examples. Few of them accumulate in the nucleus of the host plant. So these are subcellular localization studies. In protoplast experiments, so the proteins are fused to, I think, M-Sherries. That is a reporter gene system that you can see in biconfocal laser scanning microscopy and also under the microscope. And here are candidates which accumulate so the fungal protein accumulates in the nucleus of the plant. And the important question is, Does this have any meaning? Does this have any effect on the performance of the plant, or does this protein manipulate actually the expression pattern of the plant? So we did several constructs, I don't go into details, to really investigate this protein functions. We used here the hairy root system initially and pre-characterized the identified effector proteins, and ultimately we took the effector genes, the fungal genes, and transformed them stably into Arabidopsis plants and we checked then whether this has an effect on plant growth and development. And in fact, many of them have. I show you a few examples without really knowing what that means, but that shows you already that there's a strong influence of these fungal proteins in Arabidopsis and potential biotechnological applications. Here's one effector protein. If you overexpress this fungal effector protein in Arabidopsis, they are small. So apparently this effector protein inhibits somehow growth of the plant cell or plant. And it might be, so it's not what we are looking at or what we want to have in our hand, but we know that something is happening here in manipulating plant growth and performance. But we immediately found that bonsai people are highly interested in that. There are other effector proteins like this one, which relatively strongly affects the redox capacity of Arabidopsis plants. Actually, this effector protein stimulates the expression of glutathione S-transferase subunit U, a downstream enzyme, or the gene for a downstream enzyme that is influenced in the ascorbate metabolism. Another effector protein is involved in salt stress tolerance. I'll show you a better example. And we found two effector proteins which interfere with the defense mechanism. Actually blocks the salicylic acid defense pathway, activates a jasmonic acid defense pathway. That means it's specifically upregulated when the plants are attacked by necrotrophic microorganisms, bacteria or fungi. So basically, when we overexpress these two effector proteins in Arabidopsis, they are more resistant against necrotrophs, but not biotrophs. And what could be maybe interesting for you could be this slide. Here's a guy. Actually, you probably don't need to know the number because it doesn't tell you anything. This is an effector protein that confers osmotic stress tolerance. Actually, the plants were growing on 2%. The seedlings were growing in 2% PEG, 6,000 for 7 days. And you can see all of the control plants die. There are different control plants, empty vector, different other effector proteins not related to that. But this one here has an effector protein from Piriformospora indica, which somehow confers PEG resistance over a period of at least 7 days. So that can be for sure be interesting for biotechnological application, as well as understanding what's really going on in plant microbe interaction. I have a few more minutes. This story is very short, and I would like to touch two more aspects. So I hope I could show you that there is an enormous potential for effector proteins to identify the function of these effector proteins. In each of these endophytes, there is, as far as we know, really nothing that they have all in common. So you cannot predict the function of an endophyte from one organism to another, one effector protein from one organism to another. So there's a huge potential for further investigations. We shift gears now to another endophyte, Mortierella hyalina. And Mortierella hyalina has a garlic-type smell. So the smell from garlic is because garlic releases volatiles. These are about 130 different volatiles. Most of them contain sulfur, and that is the characteristic smell that we all know from garlic. This is comparable, not identical, but comparable to the smell that you can have when you grow Mortierella hyalina on agar plates. Yeah? But this is not caused by a mixture of different volatiles. There's just one volatile. Just one volatile. Let me cut that down a little bit. One volatile, sulfur-containing volatile that is released by Mortierella hyalina and perceived by the leaves of the plant. We identified the structure. This is the structure, a quite simple molecule, but you cannot buy it, so we can synthesize it. It has a central C atom and then 3 sulfur atoms, and more than 90% of the volatile composition that is released by Mortierella is this compound. And in fact, it's very interesting. That is promoting growth. When you use only this volatile, apply it in petri dishes to Arabidopsis or also tobacco seedlings, you see a growth promoting effect. Here's 10 microgram, here's 100 microgram. Larger concentration or higher concentrations show inhibitory effects, but don't forget these are covered petri dishes. The volatiles cannot escape, so in nature this does not have any meaning. Basically what it only tells you, if there is some volatile available compared to the control without the volatile, we have some growth promoting effect. The reason is the volatile is taken up by the leaves, and I briefly want to summarize what comes out of here. That is the interesting aspect of considering growth promotion. So what is happening with sulfur in plants? There are two directions for what sulfur is actually used. And there's a branch point for this sulfur metabolism. The first part of the sulfur metabolism is sulfur-containing amino acids, of course, everybody knows. And then if you have more sulfur-containing amino acids and they are rate limiting, they stimulate the primary metabolism that ultimately can lead to growth promotion. The second part are defense compounds. Sulfur-containing defense compounds. And the Mortierella compound actually triggers directly after the branch point into the stimulation of this amino acid and primary metabolism pathway, which tells you that this is a nice component to actually stimulate the primary metabolism, and consequently we can see growth-promoting effect. So take-home lesson from this is there can also be volatiles released by the microorganism, which result in the stimulation of plant performance. Let me briefly then touch the last, if I still have 2 or 3 more minutes, the last aspect that I would like to touch, and that these are hyphal networks that connect different plant species. And I think it's very, very, very important if you look at ecosystems, very important if you look at agricultural areas. The plants do not grow separate individually. They don't have an own mycorrhizal symbiotic area. They are all connected to each other. And this, as you all know, results in information and nutrient exchange, but nobody really knows how that is happening. And nobody knows how important that is for agriculture. And we can actually use Piriform mospora indica for establishing such a hyphal network system in the lab to really see what's happening. So I don't want to go into all the details. I don't describe to you the experimental setup. It's all in petri dishes, and then we have readout and infection processes. I just show you the summary slide. So this is the system that we are using in different ways, and this is the primary one published, so I can show you the summary. This is one plant. This is the neighboring plant. So this plant, the leaf of this plant, will be infected with a very small drop of a necrotrophic fungus or bacterium, doesn't matter. So one leaf gets 2 microliters of a bacterium or fungus. Yeah? And what we actually check is the leaf of the neighboring plant informed about this thread here. And is this dependent on a connection that is established between Piriformospora indica hyphae? So what's happening? So what we found out, the thread information is traveling to the roots in form of jasmonic acid. So a defense hormone that travels to the root and takes between 50 and 60 minutes in our system. And then we have, of course, all the jasmonic acid response here. So genes are upregulated, defense compounds are synthesized, jasmonic acid level is upregulated, and so on. Everything that we are expecting if the signal travels down. And now the signal has to be transferred to the neighboring root via this hyphen network of Piriformospora indica. And during this traveling, some of the information is lost because the hyphae don't understand jasmonic acid signaling. They don't speak the plant language, so they don't know what jasmonic acid is. But they realize there's something going on here. Some thread information is reaching me. So what they actually do is they transfer some information to the neighboring plant, and here is no specificity of this information, but be careful, some thread information. And this thread information is then decoded in generating or synthesizing higher level of abscisic acid, and this higher level of abscisic acid travels then somehow to the leaves of the neighboring plants, and for instance one response that you can see is closing of the stomata. So what I would like to say is, when you work with endophytes, when you look at endophytic fungus on individual seedlings, plants, or in your field, don't forget that these hyphae connect neighboring plants and transfer information from one plant to another. And that can be very important. And you know that it's not only information, it's also carbon, it's also phosphate that can be transported from one plant to another. So you have a huge network in your field so that the information is distributed, and this is probably something that is not really understood. We have now set up a huge system in which we can test different hyphae, different threads, different signaling compounds. We have nice readouts in which we can measure the response in 50 minutes because we have fluorescence markers to show what's happening. So we can now try to understand how the signal travels from here and how this is converted in the hyphae and how that can induce a response in the neighboring plant. Talk about this next time. These are summary. This is a summary of what I wanted to talk, the information that I would like to transfer to you. So, endophytes have different tools to manipulate plant signaling and metabolic processes. So different chemicals, different peptides, everything might be highly specific for a particular fungus. But the plants can respond to these signals only with their established pathways. So chemical compounds probably induce in all systems very rapid calcium elevation, and this calcium elevation is necessary for the symbiotic interaction. And one of the earliest events is phosphorylation, which tells you which compounds are then activated or inactivated. Very important is that the information is systemically transferred within the entire, or distributed within the entire plant body, in particular from the root to the leaves. I didn't talk about a compound which we identified to be involved in this systemic transfer. So resistant genes may control resistance against pathogen attack, and Piriformospora indica may control the expression of these resistant genes, may upregulate the expression of these resistant genes, and thus confer better resistance against pathogen. And this again is a systemic effect. The information travels from the root to the leaves. There's a huge amount of effector proteins in all of the different endophytes that somehow manipulate plant performance, but we have no idea what's actually happening and what is the role of these effector proteins. Volatiles from the microorganisms can be involved in promoting plant growth. I didn't talk about nitrate-fixing bacteria. And hyphae connections might be very important in an ecosystem, also in an agricultural system, to transfer information as well as carbon or phosphate from one plant to another. And I'm a little bit late as usual, but hopefully it was a little bit interesting for you. I hope you enjoyed the talk and have some ideas how we can communicate, how we can interact, whether we can bring together basic plant physiology on endophytic plant microbe interaction with agricultural application. Thank you very much for your attention.
And if anyone in the audience has a question, I think we should be free to ask the questions immediately. I have a question, Ralph. I'll start off.
Sure.
Knowing that I'm going to give the talk next. You mentioned about calcium signaling being elicited when P. indica infects Arabidopsis. What about plant pathogens when they infect the rhizosphere?
I think it's the same mechanism. Also there you find calcium elevation.
Yeah.
The specificity is what is happening downstream of calcium in the plant cell. So I showed you one example for the chitin. Chitin is a pathogen-associated molecular pattern, so a pathogen that releases this chemical, and that also induces calcium elevation, genetically different from beneficial one, and that induces downstream of this calcium elevation very strong defense response. Genes, metabolites, signaling events, actually only defense response. MAP kinase 3, 6 signal, everything that activates the defense. And the cellotriose, and this is an example for a beneficial fungus, is actually not involved in that. So the compounds are different, the signaling events are different, the phosphorylation pattern is different, and it leads primarily to the stimulation of enzymes which are involved in primary metabolism. So I think the calcium signal is a very nice readout, but it's genetically different. Yeah, has nothing to do with each other. One in the pathogenic leads to defense, and the other way, in the beneficial way, it leads to somehow stimulation of the metabolism, resulting in benefits for the plants.
Thank you.
Is my view. I mean, people can question that, but all the results that we got in the last 20 years support this.
Sure.
I had a question connected to poly-A-specific ribonuclease activation. So, does it lead to any hormone synthesis degradation of this poly-A thing, or something else or cell cycle is something getting triggered? Is it a general mechanism which happens? So,
okay, if you give me 2 hours, I can answer the question completely. If you want a summary, yes, it affects. Is what is happening downstream of calcium in the plant cell. So I showed you one mRNA and one protein disappears. Not all mRNAs and not all proteins. There's some specificity, and one of the candidates that we are looking at, and the likely candidate is the protein involved in abscisic acid signaling, and it's necessary to balance abscisic acid, and we see also higher abscisic acid levels in our mutants. And we know that this is one very specific protein that is missing is absolutely necessary for systemic signaling. It's a very interesting protein. I shouldn't tell that to you. Don't tell it to anybody else. So you can colonize the roots, Arabidopsis as well as tobacco roots with Piriform Mospora indica. Nothing much is happening. For 72 hours later, this SNRK2.8 protein, an abscisic acid signaling protein, is specifically and at least tenfold upregulated in the leaves. Only this one. There are 12 other members of the same gene family which do not respond to it. So it only responds to systemic signals from the roots to the leaves and is upregulated, and it does not respond if the fungus itself is there. So, make the whole story short, I think this polyribonuclease specifically controls one message for a protein that is essential for systemic ABA signaling.
So then probably some regulatory elements are there in mRNA. Maybe that would be the key thing to find out. So
right.
Thank you very much for a very nice talk. Regarding Piriform Mospora, there's a lot of similarity with the arbuscular mycorrhizal fungi. It's reported that both of them performs almost the same function, like taking up of the nutrients and other things. Now, when we talk of the communication between the two plants within the system, does the signal get lost when here you are doing it in the lab studies, but when you're doing the same thing in the field?
Good question.
Can there be a loss of the signal between the two?
Good question. We didn't test that. Of course, that is for sure absolutely necessary to test. So we did only experiments in the lab to understand the molecular basis of this information transfer. Whether there's any relevance for ecosystems or agricultural application, and whether this also functions in the field needs to be tested. We didn't do that.
Thank you.
Yeah, I have one query. You seems to be thinking only hormones like jasminic acid, but in your transcriptome data or one of the slides you have shown BAC1 protein, which is steroid related signaling. So do you have some information in that direction as well?
Yeah, so steroids appear to also play a role in stimulating lateral root initiation in our system. The problem is we don't know much about the function of these hormones. So if you go to the literature, it's described as very complicated. It influences everything, can upregulate everything. So that makes it very difficult for us to really pinpoint down what is target and what is really happening and why is it upregulated if everything is described. So it plays a role, we know that. We didn't determine the concentration of the hormone. We can only see that some of the steroid responsive genes go up specifically in the region where lateral root development is initiated. How that is related to the signaling process, we don't know.
Thank you. I think we should stop. Yeah. Let's thank Professor Ralph for his excellent talk. Thank you, Ralph.
Thank you. Thank you, Professor Ralph. I request Dr. A.K. Saxena, Director ICR, National Bureau of Agriculture, Important Microorganisms, SMO, to hand over a memento to thank Professor Ralph.
Thank you, Ralph.
So now we have the invited talk by Professor R. Umashankar, Department of Crop Physiology, US Bangalore. I request Dr. N. Iranna, the Symposium Coordinator, to welcome Professor Umashankar with a brief biodata.
Ralph, that will hinder your vision. You may want to come back.
Friends, it is my pleasure to introduce Dr. Umashankar. Professor Umashankar is an ICR Emeritus Scientist, Department of Crop Physiology, US Bangalore. He is an elected Fellow of Indian Academy of Sciences, Bangalore. He was a Fulbright, even today he is Fulbright Fellow at the University of Massachusetts, Boston, and Honorary Fellow of the Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. He has published more than 230 peer-reviewed papers, about 30 articles in books and monographs, besides authoring or editing 15 books. He serves as an expert member on the advisory panel of the Department of Science and Technology, Department of Biotechnology, Council of Scientific and Industrial Research, CSIR, Government of India. And Professor Umashankar is recipient of Sir C.V. Raman Young Scientist Award and State Environment Award from the Government of Karnataka. He also served in the editorial board of many scientific journals as Current Science, Biotropica, Journal of Plant Physiology, Journal of Biosciences, etc., etc. His research contributions over past 2.5 decades span the area of plant evolutionary biology, conservation biology, and bioprospecting. His group has identified many endophytic fungi and bacteria which are capable of modulating plant growth and developments under stressful conditions. These endophytes are now being used for technology development. So, please, sir.
Thank you. So thank you very much, Irana, for the introduction, which I think I don't deserve. And thank you all for staying for the talk. I know we are fast approaching lunch, and it was, I think, the nefarious design of my good friend Nataraj Karba to sort of crunch me in the next 20 minutes to sort of finish my talk before lunch. Hopefully I'll do that. We just heard an excellent talk by Ralph on looking into the molecular mechanisms of plant endophyte interactions. And I believe the next 2-3 days we shall be hearing more of these stories from many of the participants who have now come here. I will now take a different look at this entire business of plant microbiome in the context that I thought might lend a perspective to at least the younger researchers who are assembled here today. And for that, I'd like to introduce you to this huge project that has been launched by the National Institute of Health called the Human Microbiome Project. And this is probably the next, I would say, biggest project that the NIH launched after the Human Genome Program. Now, this over the last decade or so is arguably the most heavily funded program in the United States and also for matter globally, and it has funded lots and lots of research, of course, centered about the discovery about human microbiome. And from these studies over the last decade and maybe a few years, there have been many, many big wins, and many of you might have also read this in different literature, especially in leading journals, be it Nature, Science, PNAS, and so on and so forth. Now, some of the big wins that I came to appreciate and also be shocked is probably one of this, that after cataloging the microbiome that populates you and me, it was estimated, and it is debated, but still it's accepted, that of about 100 trillion cells that we may have in our body, we can claim only a tenth of those cells to ourselves.
90% of the cells that dot our body is not ours. And so there's a book that writes, are we just 10% Uma Shankar or 10% human? The rest of the 90 is not me and of you that you claim to be. This also has traveled into the consciousness of the genome that we harbor in our body. Again, it's a debatable proposition that you may have 21,000 to 23,000 or maybe max 30,000 genes in a cell. But for these 30,000 genes in a cell, you are far, far outnumbered by the microbial genome that is in each one of the cells or around the cells doing whatever they are doing. And so by number, we are far, far outnumbered by the microbial genome. And then out of this human microbiome project, there have been some very elegant findings which are made into all sorts of press, and one of those is this business about linking microbiome to obesity. And this is a classical study published in Science which shows that how the members of a pair of a twin, their obesity, in this case this person, and that is non-obese partner of the twin, their obesity could be ascribed to the gut microbiota that this lady has in her gut compared to that. And this was now demonstrated very clearly by showing that if you now take the gut microbiota of this lady and put it into a recipient mice, this recipient mice becomes increasingly adipose. Whereas if you take the microbiota of that lady and put it into the recipient mice, the mice remains lean and healthy. Now this was actually a great, I would say, shock to know that microbiota, the gut microbiome, can actually spell what is your overall health status, in this case the obesity situation. And this is more cliched. A number of papers have been published, but this is a paper which I picked up just last week from Nature, September 2019, where they show that our microbiome, at least in the first few days or weeks of infancy, depends completely about how you were born, how you were delivered. And if you were delivered by C-section, as the story shows, the C-section babies, that is the blue ones, are completely different in the microbiome compared to children born through the vaginal delivery. And of course, over the days of infancy, they seem to get mixed up, but the early period of infancy is completely determined by how one is delivered. This may have implication about or on the immune competence that the children have that are born by either of those methods. And today, thanks to those basic discoveries in the Human Microbiome Project, we have something very hilarious, but also in practice. Just last week I was in Pune and a recipient of what is called as a fecal microbiota transplantation, where this particular patient was a patient of a very, I would say, quite a difficult disease called IBS, or syndrome called IBS, irritable bowel syndrome. And all that the patient had to do was ask his spouse for a little bit of, not money or love, but just a little bit of stool sample, which is now sort of purified, and that sample is taken by colonoscopy, and this person actually gets relieved of his irritable bowel syndrome that modern medicine could not have medicine for. So this is a very, very practical application of how the study on microbiome has actually been a relief in some of these diseases. But more than all of this, in terms of its pervasiveness, to me at least, has been the ideology that in the last decade, ever since the NIH launched this project, there has been a massive, I would say, change in a perspective on microbiome becoming the new fashion, and leading labs across the world have started looking at questions on microbiome. Just the other day, just day before yesterday, there was a very interesting paper about how microbiome can be related to the mental health of people, and how in psychotic treatments you can actually introduce microbiome as one line of therapy other than the usual therapy that are normally now provided in mental illnesses. But as a geneticist, For me, something more important is this conceptual framework, which I would say is a paradigm shift in what we have been thinking before, let's say about 5 or 6 years before now, and that is the following. Now, we have been thinking about each of us as a genome. It could be a plant, it could be an animal. But today, thanks to the unraveling of the overarching influences of the microbial genome, we know that the microbial genome, which is closely associated with us and in many, many ways influences our own sort of physiology, behavior and whatnot and development, as probably Ralph mentioned a while ago, you have these very intimately sort of intertwined, and so the microbial genome and the host genome actually go hand in hand, and to that extent people have now called that it is better to now take both of these, the host and the microbial genome together, and refer to them as the hologenome. And what we are decided by is the hologenome, the genomes of the host and the microbial genome, which interact with the environmental metagenome. And so conventional Mendelian population genetics tells us the following, and that is that if you want to decompose the phenotypic variance of any organism, it is classically defined as genotype by environment interaction, where the genotype refers to the genotype of the host. But I think it is no longer so, because you now have a new enterant which we had not yet identified, only recently, and that is that the phenotype is now no longer at the hands of only the genotype of the host, but it is a genotype of the host interaction with a genotype of the microbe and that of the environment. So imagine, therefore, you now need to write all your epistatic interactions differently, your over-dominance interaction differently, your additive interactions differently, because you now have to include how does this interact with this in an epistatic manner or an additive manner or in a dominant manner. So, but that is for yet another day. Now, having given this introduction on the human microbiome, the point that I wanted to raise, and in a public platform such as this where it is open for us to now debate, is that viewed in the context of the human microbiome, plant microbiome seems to be a poor cousin of the human microbiome's work, simply because it hasn't gained, it hasn't attracted the attention, the banner headlines like what we have had in the case of human microbiome. We have not made it to this pages and splashes of newspapers and general TV headlines simply because we are less sexier than animals. I'm talking about plants. And when I give such a talk, just to force this in, I believe sometimes I'm also confused about who I am. And so when I give an authorship for this talk, I famously now put this across that this authorship of my talk henceforth should now be called just not me, but the microbiome of myself who is talking 90%. 10% is me, isn't it? So you now actually go into a daze when you really get hit by this business of microbiome overwhelmingly ruling our behavior, unknowing to you that it is actually into you, over you, driving you and whatnot in terms of its ever-reaching and overarching effects. Now, against this backdrop, we know lots and lots of things about the gut microbiota today. You go to any journal, you will know that there are lots of work on the metagenome of the gut microbiota. Probably we also know the functional relevance of much of the microbiota. But go to literature and look at, do we know enough of the microbiota, the microbiome that is inside a seed? We hardly know anything about the microbiome that sort of populates the seed. Just what are those microbiome doing in the seeds? Recently, very recently, a student of our lab looked at finger millet, that is ragi, Eleusine coracana. And if you take any seed, single seed of ragi here, finger millet, you'll find that that is dotted, not on its surface. You can surface sterilize the seed, but inside the seed, when you look at, you will find that that seed is populated by huge amount of microbiome, in this case bacterial microbiome, bacterial endophyte if you have to use the word. And imagine a situation where you have all these metagenome of the bacteria lined up within each one of those seeds of finger millet. What are these doing? What are their functional roles? Can we now understand something and crack some of that to try and see if we can understand the interaction that they have with the host genome, in this case the finger millet genome? Well, we all talk about endophyte. Ralph has talked. Many of us are going to talk about endophyte in the next few days. But to me, I feel we still haven't understood yet completely. In fact, we have not even touched, I would say, the surface of it in terms of asking what has been the origin and evolution of endophytes. It's enigmatic. And I stress this word enigmatic simply because I would like to provoke here that unless we understand the origin and evolution of the endophyte, it appears we will be so much so poorer in trying to apply endophytes, whether in medicine or in agriculture. And that's a limitation I'd like to sort of flag in the course of my talk now and maybe later. So it is vital to understand the evolutionary significance of what these endophytes are doing. It's fine to talk about P. indica doing this or that, but what is it doing to be able to understand how can it do better and how can we now use it as a handle to crank a little bit more out of the P. indica manifestations on the phenotype of a plant. So, against this backdrop, very quickly, let me tell you 3 or 4 stories as Ralph made it out in his own lecture about what we have been doing at GKVK in the University of Agricultural Sciences over the last few years. And what we have been trying to look at is, we have been trying to ask this question about what are the roles of microbiome in 3 or 4 processes, and I'll just give you one example for each one of them just to illustrate the sort of work that we have been doing. One will be on seed development, another on early seedling growth, graduating from seed to seedling growth, and then of course what we have been talking about, modulating plant responses to abiotic stress, and in secondary metabolite production. Actually, the work in GKVK started not in this or this or this, but we have been working on this business of secondary metabolite production for quite many years, but I will relegate that to just 2 slides because that is not the most important thing I want to now talk this morning. Now let me take you through each one of them, just 2 or 3 slides, hopefully finish on time. Role of microbiome in seed development. Now as physiologists, we all know that every plant now brings in its resource or source from the leaf where the assimilation happens to a growing sink. It may be a tuber, it could be a seed, and so on and so forth, by well-recognized methods of phloem loading and phloem unloading. So the phloem loading happens into the phloem, and then the phloem unloading happens from the phloem into the sink, and that's how we get all the seeds. We understand pretty well today the physiology, the molecular basis of this phloem loading and unloading. The question that we asked is, can we now ask if the microbiome has any role in this business of loading and unloading of assimilates into the seed, and if so, it has a natural consequence of whatever application that we might think of. So one of the ways of addressing an extreme question like this is to apply an extreme sort of method. And the extreme method that we applied was, and this trick I learned from my friend Dr. Suri Narayanan, who's sitting here. I went up to him one day and asked him, how do we get rid of endophytes? Now I'm going to use endophytes and microbiome synonymously. How do we get rid of endophytes from a plant? He told, just apply systemic fungicide to the plant that you're worried about. So what we did was, we took this plant called cowpea, Vigna unguiculata, and rolled up those pots with cotton swabs around them, and swabbed generously Bevestin, which is a very common systemic fungicide used by lots of people in agricultural research. This easily gets into the tissue, goes all through, and then knocks off everything possibly there which is sensitive to this fungicide. And then asked, what is the fate of the seeds that are developing within each one of the pods which are treated by the systemic fungicide? Now this was a shocking result that we got, and the results showed us that seeds that are treated or seeds that are got from pods treated with the systemic fungicide have on an average about 15 to 20% reduced seed weight compared to seeds that are coming from pods that are not treated with the systemic fungicide. So all that has been done is they have not been treated. Here the pods have been treated with systemic fungicide. So you end up in a colossal loss of about 15 to 20% of seed weight just by treating the pods with systemic fungicide. The question is, of course, too tempting to ask, is it because of washing away this endophytic fungi that may have been laden in the seed during the seed development process? There are a little bit of debatable issues here, whether there are collateral issues that is now affecting the seed during the development process. I won't have time to answer that. I can take that question after maybe my talk. So at this moment, if you now accept this, it is tempting to conclude that the seed microbiome seems to play a significant role in the mobilization of resources into developing seeds. Very recently, after this paper was published, we entered into some sort of discussion with a colleague of us at another institute in Delhi who told us that it's possible, and there are some already indications that endophytes might play a role distinctly different from pathogens in this manner that you could expect the endophytes to now upregulate what are called as sweet transporters, which are membrane bound, which now help transport the sugar from the symplast into the apoplast where all of our endophytes are swimming. And these, therefore, when they are able to now upregulate the sweet transporters, can now get in more and more. They can actually suck out more and more sugar out of your symplast, that is the leaf, and put it into the apoplast. That, as a spin-off, can be now amount of sugar that can be unloaded into the sink, and so you have a win-win strategy. Your endophyte gains carbon, also this developing sink in the seed, in the fruit, in the plant also gets the same carbon. This is too early days to now show whether it is true or not, but already the experiments are on cards to now see if this is indeed working or not. The second is the role of seed microbiome in seed germination as a graduation from the development of the seed. All of us know, as plant physiologists and as seed technologists, that for the seed to germinate, one of the most important enzymes is alpha amylase that mobilizes the starch by breaking the starch into reducing sugars, which powers the ATP required for the radicle and the plumule development or shoot development. So, again, the same question, is there a role for microbiome on a gross scale? And so in this case, what we did, we answered the same question using paddy as a system, where we treated paddy with a systemic fungicide, again in this case Bevestin, at a concentration which is not toxic to the plant, which is 0.2%. All that we do is put the seeds, treat the seeds in this fungicide for about 3 hours, take it out, wash it, and then do the experiment. And what is the experiment? The experiment is simply germinate and grow the seedlings of paddy. What you see in this graph is something, again, spectacular, and that is the following. You have these paddy seeds or seedlings that have come out of paddy seeds that have not been treated with the fungicide. And those are paddy seeds exactly similar to this, except that those seeds have been treated with the fungicide. And what you find here is about roughly 40 or 50% reduction in radical length in seeds that are treated with the fungicide. It's too tempting to now suggest that this reduction that you are getting here is probably because you have knocked off the endophytes from these plants by the treatment with the fungicide. So is it true or is it not? What could be done? What was done is, first of all, we looked at whether these roots have endophytes in them. So sections of these roots were taken, plated, and we found that profuse endophyte emergence were present in any part of the roots that we care to now look at. Now, if you do the same thing for this one, this plate is as clean as anything that you can get. There's absolutely no endophyte emerging from these seedlings that are coming out of seeds treated with the fungicide. So that's as good as that. Now the question then was, if we can now take these endophytes, a consortium of them coming from these untreated seedlings, and put them into this one, can we revoke and restore back the growth as a complete circle to demonstrate that, look, the reduction in the seedling growth that you've got here is perhaps due to the loss of these endophytes in these seedlings. And so we did that, and what you find is that indeed you can recover the growth of the seedlings. Once you now take the endophytes from here and put them into there, you get a growth back, which is nearly the same as that, much better than this, though not as good as the control, but nearly the same as that. This shows that you probably are at a position to demonstrate that the totality of endophyte that is there within the seedling might in some way be contributing to the overall development of the seedling, which we probably have not realized yet. Now, what is happening, how is happening, etc. are all stuff to be now worked down by someone who is interested in this. This is now published somewhere. I would like to mention this, show this slide because Dr. Karwar is here. Now he did and demonstrated this, but in a bacterial system using streptomycin, and shows exactly the same result as us, that we used bevestin to knock off the fungus. They used streptomycin to knock off the bacteria, the endophytic bacteria. Okay, so there are many questions. I will not spend time on that since I can't answer that. Okay, 5 minutes more, Nataraj. The third area that we are looking at is the role of microbiome in imparting tolerance to abiotic stress. Now, this is something that has held our fancy because this something is very close to application, and as Professor Saxena mentioned this morning and Ralph mentioned this morning, I think this is something that we are all keen at the university to now do something about. So our target was to build up a library of endophytes from different plants based on a rationale. Some of you would have heard about this habitat adapted symbiotic theory by Rodriguez and his group. So we went around different parts of the country, from coastal peninsular India to the Thar desert of India to the cold desert of Himalaya and to the wet subtropical forest of Arunachal Pradesh, and got lots and lots of these fungus from these places. And the question is, screen them out and do some work to see if we really can do a short-term sort of response study on these fungus in terms of ameliorating the tolerance of crop plants to stress. Here's one example from Thar desert. Many plants were surveyed, and lots of fungus were pulled out. And this is the same Thar desert from where P. indica Ralph was pulled out by Amit. And here is a fungus called LAS6, which is tolerant to high temperatures in plate culture. And when you now put this fungus into paddy systems, you find that paddy system that is colonized by this fungus does much better at an elevated temperature compared to those which are not treated, indicating that there is some degree of ability on the part of the fungus to now confer the tolerance that it has itself got into a host plant that it has not ever reached up to. These are not fungus from rice, it is from some other plant growing in the Thar desert. So today we have lots of these systems. I'm just putting this one example. We have lots of the systems, be it for high temperature, be it for salinity, be it for drought, be it for oxidative stress, and very soon for pH stress. These are material that we are generating from all and sundry places that I showed you, but the hope at the end of the day is to ask what is happening in many of these responses. So we have now started some work on asking, can we now look at starting from the transcriptome, look at what are the genes that are getting upregulated, do we make any sense of the functional relevance of these genes that are getting upregulated, and these are work under progress. And one of our postdoc actually is now working on this Thar Desert material to look at what are the differentially expressed genes and can we now functionally validate them to now demonstrate that these are the reasons why you have a better phenotype in plants that are colonized by some of the endophytes coming from the Thar Desert. Last, which was the first work that we started in the lab here, is the role of microbiome in plant secondary metabolite production or secondary metabolite production. And this was a science paper published in 1993 by Gary Strobel, who showed that from Taxus brevifolia, you of course have this compound called paclitaxel or Taxol, which incidentally is the largest anti-cancer metabolite being sold in the clinical market. And will not go into the reason and the mechanism of action of this one. But this guy, incidentally, Karwar worked with Gary, showed that if you now take endophytic fungi from this plant, these fungus, many of them, including what Ralph mentioned about Taxomyces andreniae, is able to now produce paclitaxel in broth culture, and that became, I would say, the sensation. I would say the sensation because you now demonstrated that you can now get paclitaxel from a fungus in shake flask. You don't have to now depend upon the yew tree anymore. That was in 1993. And after that, what happened? Lots of foot soldiers around the world started emulating Gary Strobel, and our lab was one of those labs. And we started working on many, many metabolites. I'll just flash two to give you to tell you that we also do something. And so we looked at yet another important anti-cancer molecule called camptothecin, and we were able to show that many fungi isolated from a plant that produces this molecule camptothecin. Incidentally, this is the third largest anti-cancer compound sold in the clinical market. When you pull fungus out of these plants, you find that the fungus makes the same compound as the plant does. So again, you have lots of excitement and all the stuff. And just a popular excitement, just a couple of years back, intern came to our lab and he showed this one, that many of the girls who know about this plant, Lawson and Lawsonia inermis, produces this dye called Lawsone, which you use for this painting the hand. You can now pull out fungus from this plant and show that it produces a pigment in the petri dish. So you don't have to actually go to the plant. You just put your hand in the petri dish, you get whatever pattern you want. Okay? Just to demonstrate that these things are possible. But let me quickly end. I am sick of this because you can actually demonstrate everything you want. There was this paper from IIM Jammu who showed that a strain of endophyte called Alternaria alternata produces capsaicin from capsicum, of course, and so you don't need chilies anymore. You sprinkle a little bit of that fungus and that is enough because it has got capsaicin. There are many, many stories. After working for a long time on this area, we asked this question last year, exasperated because we don't have any answers on any of those questions, and the paper had this title, actually I wanted to use this word in the title, how the hell, okay, it already came out of my mouth inadvertently, and of course I didn't put it in the title, how and why do endophytes produce plant secondary metabolite? It was later told to me, I got very severe criticism on this paper, but thankfully it was accepted, and after it was accepted, this gentleman comes to me and tells me, so congratulations. I asked him, what about? He told, I reviewed your paper. And that gentleman is Suri. So this is a small world. Okay, so in summary, friends, in summary, you see just seeds. Well, that was not my line. This was a cartoon that I sort of bought out from the Google. I didn't have any way to change it. But then I added this footnote, but I see the microbiome also. that sort of gives a good thing to that. But more than that, more seriously, and as a sort of starter for this entire symposium, from my perspective, I think we have just seen the tip of the iceberg in the area of endophyte biology. There are many more questions than we have answers for. In fact, Ralph's 2018 paper in Frontiers, this is not copied from you, Ralph, has a title ending with this title, More Questions Than Answers. But as we seek answers, can we look at some applications in agriculture? Because till eternity we can look at answers, isn't it? But as our Vice Chancellor pointed out, maybe in the short term we need some solutions. And people like Professor Saxena, maybe Pius Thomas, probably will not, because he has a very sort of unpredictable way of contesting whatever I say. May agree with me that probably in the short term we probably need to also look at applications in agriculture. This was a beautiful paper. Whenever a beautiful paper comes, I feel I should have written that paper. And because this guy did not do anything, all that he did was he collated all the papers in endophyte biology so far and did a meta-analysis of the paper, about 180 odd papers, and did a meta-analysis to show that and answer the question, do endophytes promote growth of host plants under stress? And the short answer he showed was yes, it does. And this is geography across, plants across, and on 3 stress. A is drought, B is salinity, and third is mineral deficiency, N deficiency, nitrogen deficiency. And I believe this sort of a meta-analysis gives us the conviction that endophytes may finally have a promise of its application in agriculture. And towards this end, while we ask all the questions, while we clean up the mess, there have been some initiatives in our labs where we now are trying to do seed and seedling biopriming. And I've spoken to Professor Saxena when I visited his institute last time, where simple treatment of seeds with certain fungus that have proven in experiments are taken to pot cultures, where they again are sort of amplified in terms of the phenotype, and taken to the field to see if we can have some answers to questions in the field or on the field in terms of salinity tolerance or drought tolerance and so on and so forth. This was a very interesting paper published in Trends in Plant Science, and the title of the paper was Plant Breeding Goes Microbial. I was trained as a plant breeder, and I believe today I have a transformation done here by some of these demonstrations, that if you can now stably, when I say stably, from the use of the word microbiome, if you can vertically have a microbiota transferred from the plant to its progeny with a stable inheritance of the phenotype, you've actually achieved a microbial method to plant breeding. And so that's probably the way forward. So I'd like to thank a lot of people from the lab who contributed to many of the studies that I've sprinkled across in my talk. All of the work, the snippets at least that I've showed you, were published, were supported by Department of Biotechnology and the current ICR CAST project. And finally, thank you all for listening, and we are 10 minutes late. Thank you very much. I don't think we should entertain any questions at the cost of missing our lunch.
Do you have any questions? Quick question.
The seed microbiota, is it always coming from the soil and seed, or is it
both roots? One is vertical, that is from seed to seed, and another is horizontal.
From the air also, is it possible?
This air also possible. Suryanarayan would further testify on that. For many of the monocotyledon like grasses, most of them are vertically transmitted. But in dicots, the vertical transmission is not as much prevalent as is the horizontal acquisition of the microbiome from the environment. It could be from air, it could be from soil, from the litter. I think he will probably talk a little bit more on that in his afternoon talk, maybe.
Just a small query.
Yeah, please.
Any attempts to multiply the microbiome of a healthy plant outside the host?
Microbiome of a plant outside the host. You mean in plates? Yeah, we are doing that.
No, as a formulation, if you have to multiply the microbiome and then give it as a formulation to.
Okay. Oh, sorry, I mistook your question. Sir, at this moment we are not yet at the formulation stage. But for example, Professor Barik is coming tomorrow. We are already talking with Professor Barik to now develop some formulations of some of the half a dozen strains, half a dozen isolates that we have got to now actually go into a formulation stage to bring it out and then maybe use it in the field trials and such as, if that's a question. Yeah.
A microbiome which has been isolated from a healthy plant, can it be multiplied, the entire microbiome, not 2 or 3 cultures? Of
No. We have not attempted at all. No. So you're asking as a consortium, as an assemblage of the microbiome? No.
Sir, an excellent and exciting presentation. Can you please throw some light on the ragi microbiome? We have 3 samples here shown.
Yeah.
What is that 3 samples referring to? 3 populations?
Those 3 populations are the 3 accessions of Eleusine coracana. So Hemapriya might be giving her presentation where she is interested in looking at the evolution of microbiome or actually change in microbiome through the process of domestication of Eleusine coracana from Eleusine africana. So she is studying the metagenome of africana and coracana and ask if there are differences. So within each coracana, we take 3 accessions as biological replicates. So those are the 3 accessions. Okay?
Thank you, sir.
Thank you.
Thank you, Professor Umashankar. So please stay on the stage. I now request Professor T.S. Suryanarayanan, Vivekananda Institute of Tropical Microbiology, Chennai, to hand over a memento as a token of appreciation. Thank you. So dear delegates, before we break for lunch, I have a few announcements to be made. The lunch is organized in the Botanical Garden of University of Agriculture Sciences, Bangalore, and it is till 2 p.m. And second, we have 2 technical sessions after the lunch, and all speakers kindly hand over the PowerPoint presentations to Mr. Tinu Thomas here. He can help us in organizing the things. And third, all the poster presenters are requested to hand over your e-posters to Ms. Pratibha, who will be near to the registration counter. Thank you.